The sugar glider is a small, omnivorous, arboreal, and nocturnal gliding possum belonging to the marsupial infraclass. The common name refers to its predilection for sugary food such as sap and nectar and its ability to glide through the air, much like a flying squirrel. They have very similar habits and appearance to the flying squirrel, despite not being closely related, an example of convergent evolution. The scientific name, Pitoris breviceps, translates from Latin as short-headed rope dancer, a reference to their canopy acrobatics. The sugar glider is characterized by its pair of gliding membranes, known as patagia, which extend from its forelegs to its hindlegs. Gliding serves as an efficient means of reaching food and evading predators. The animal is covered in soft pale gray to light brown fur which is countershaded, being lighter in color on its underside. The sugar glider is native to parts of mainland Australia, New Guinea and certain Indonesian islands, and it was introduced to Tasmania, probably in the 1830s. It is a popular exotic pet, but is prohibited in some regions, including parts of Australia and the United States. Taxonomy. The genus Pitoris is believed to have originated during the early to mid-Miocene period, 18 to 24 million years ago, then dispersed from New Guinea to Australia, where Australian Pitoris species diverged. The earliest Pitoris species occurred in Australia 4.46 million years ago, and the sugar glider is the only Pitoris species native to both Australia and New Guinea. The species is divided into 7 subspecies, 3 occur in Australia, 4 in New Guinea, although debate regarding current species delineation continues. These 7 subspecies are currently designated by small morphological differences such as color and body size. However, genetic analysis using mitochondrial DNA indicates that the morphological subspecies may not represent genetically unique populations. Contrary to the current geographic distribution of sugar gliders, 2 genetically distinct populations in Australia may have arisen due to long-term geographical isolation following drying of the Australian continent after the Pleistocene and the uplift of the Great Dividing Range by a process known as allopatric speciation. One population is found in coastal New South Wales and southern Queensland, and the other is found in northern Queensland, inland and southern New South Wales, Victoria, and South Australia. Further evidence is required to clarify if changes to the current taxonomic divisions are warranted. Distribution and habitat. Sugar gliders are found throughout the northern and eastern parts of mainland Australia, Tasmania, New Guinea and several associated isles, the Bismarck Archipelago, Louisiade Archipelago, and certain isles of Indonesia, Halmahera Islands of the North Moluccas. The earliest Australian sugar glider fossils were found in a cave in Victoria and are dated to 15,000 years ago, at the time of the Pleistocene epoch. The facilitated introduction of the sugar glider to Tasmania in 1835 is supported by the absence of skeletal remains in subfossil bone deposits and the lack of an Aboriginal Tasmanian name for the animal. In Australia, sugar glider distribution corresponds with forests along the southern, eastern and northern coastlines and extends to altitudes of 2,000 meters in the eastern ranges. The sugar glider occurs in symmetry with the squirrel glider, mahogany glider, and yellow-bellied glider, and their coexistence is permitted through niche partitioning, where each species has different patterns of resource use. They have a broad habitat niche, inhabiting rainforests and coconut plantations in New Guinea, and rainforests, wetter dry sclerophyll forest and acacia scrub in Australia, preferring habitats with eucalypt and acacia species. The main structural habitat requirements are a large number of stems within the canopy and dense mid and upper canopy cover, likely to enable efficient movement through the canopy. Like all arboreal, nocturnal marsupials, sugar gliders are active at night and they shelter during the day in tree hollows lined with leafy twigs. The average home range of sugar gliders is 0.5 hectares and is largely related to the abundance of food sources. Density ranges from 2 to 6 individuals per hectare. Native owls are their primary predators. Others in their range include kookaburras, goannas, snakes, and quolls. Feral cats also represent a significant threat. Appearance and anatomy. The sugar glider has a squirrel-like body with a long, partially, weakly, prehensile tail. The length from the nose to the tip of the tail is about 24–30 centimeters, and males and females weigh 140 and 115 grams respectively. Heart rate range is 200–300 beats per minute, and respiratory rate is 16–40 breaths per minute. The sugar glider is a sexually dimorphic species, with males typically larger than females. Sexual dimorphism has likely evolved due to increased mate competition arising through social group structure, and is more pronounced in regions of higher latitude, where mate competition is greater due to increased food availability. The fur coat on the sugar glider is thick, soft, and is usually blue-gray, although some have been known to be yellow, tan or, rarely, albino. A black stripe is seen from its nose to midway on its back. Its belly, throat, and chest are cream in color. Males have 4 scent glands, located on the forehead, chest, and 2 paraclosal, associated with, but not part of the cloaca, which is the common opening for the intestinal, urinal and genital tracts that are used for marking of group members and territory. Scent glands on the head and chest of males appear as bald spots. Females also have a paraclosal scent gland and a scent gland in the pouch, but do not have scent glands on the chest or forehead. The sugar glider is nocturnal. Its large eyes help it to see at night, and its ears swivel to help locate prey in the dark. The eyes are set far apart, allowing more precise triangulation from launching to landing locations while gliding. Each foot on the sugar glider has 5 digits, with an opposable toe on each hind foot. These opposable toes are clawless and bend such that they can touch all the other digits, like a human thumb, allowing it to firmly grasp branches. The second and third digits of the hind foot are partially syndactylous, fused together, forming a grooming comb. The fourth digit of the forefoot is sharp and elongated, aiding in extraction of insects under the bark of trees. The gliding membrane extends from the outside of the fifth digit of each forefoot to the first digit of each hind foot. When the legs are stretched out, this membrane allows the sugar glider to glide a considerable distance. The membrane is supported by well-developed bicarpellus, humerodorsalis, and tibio-abdominalis muscles, and its movement is controlled by these supporting muscles in conjunction with trunk, limb and tail movement. Lifespan in the wild is up to 9 years, is typically up to 12 years in captivity, and the maximum reported lifespan is 17.8 years. Biology and behavior. Gliding. The sugar glider is one of a number of volplane, gliding, possums in Australia. It glides with the fore and hind limbs extended at right angles to the body, with feet flexed upwards. The animal launches itself from a tree, spreading its limbs to expose the gliding membranes. This creates an aerofoil enabling it to glide 50 meters, 55 yards, or more. For every 6 feet traveled horizontally when gliding, it falls 3 feet. Steering is controlled by moving limbs and adjusting the tension of the gliding membrane. For example, to turn left, the left forearm is lowered below the right. This form of arboreal locomotion is typically used to travel from tree to tree. The species rarely descends to the ground. Gliding provides 3-dimensional avoidance of arboreal predators and minimal contact with ground-dwelling predators, as well as possible benefits in decreasing time and energy consumption spent foraging for nutrient-poor foods that are irregularly distributed. Young carried in the pouch of females are protected from landing forces by the septum that separates them within the pouch. Torpor. Sugar gliders can tolerate ambient air temperatures of up to 40 degrees Celsius through behavioral strategies such as licking their coat and exposing the wet area, as well as drinking small quantities of water. In cold weather, sugar gliders will huddle together to avoid heat loss and will enter torpor to conserve energy. Huddling as an energy-conserving mechanism is not as efficient as torpor. Before entering torpor, a sugar glider will reduce activity and body temperature normally in order to lower energy expenditure and avoid torpor. With energetic constraints, the sugar glider will enter into daily torpor for 23 hours while in rest phase. Torpor differs from hibernation in that torpor is usually a short-term daily cycle. Entering torpor saves energy for the animal by allowing its body temperature to fall to a minimum of 10.4 degrees Celsius to 19.6 degrees Celsius. When food is scarce, as in winter, heat production is lowered in order to reduce energy expenditure. With low energy and heat production, it is important for the sugar glider to peak its body mass by fat content in the autumn, May-June, in order to survive the following cold season. In the wild, sugar gliders enter into daily torpor more often than sugar gliders in captivity. The use of torpor is most frequent during winter, likely in response to low ambient temperature, rainfall, and seasonal fluctuation in food sources. Diet and nutrition. Sugar gliders are seasonally adaptive omnivores with a wide variety of foods in their diet and mainly forage in the lower layers of the forest canopy. Sugar gliders may obtain up to half their daily water intake through drinking rainwater, with the remainder obtained through water held in its food. In summer they are primarily insectivorous, and in the winter when insects and other arthropods are scarce, they are mostly exudativorous. Sugar gliders have an enlarged cecum to assist in digestion of complex carbohydrates obtained from gum and sap. To obtain sap or gum from plants, sugar gliders will strip the bark off trees or open boreholes with their teeth to access stored liquid. Little time is spent foraging for insects, as it is an energetically expensive process, and sugar gliders will wait until insects fly into their habitat or stop to feed on flowers. Gliders consume approximately 11 grams of dry food matter per day. This equates to roughly 8% and 9.5% of body weight for males and females, respectively. They are opportunistic feeders and can be carnivorous, preying mostly on lizards and small birds. They eat many other foods when available, such as nectar, acacia seeds, bird eggs, pollen, fungi and native fruits. Pollen can make up a large portion of their diet, therefore sugar gliders are likely to be important pollinators of Banksia species. Reproduction. Like most marsupials, female sugar gliders have 2 ovaries and 2 uteri. They are polyestrous, meaning they can go into heat several times a year. The female has a marsupium in the middle of her abdomen to carry offspring. The pouch opens anteriorly, and 2 lateral pockets extend posteriorly when young are present. 4 nipples are usually present in the pouch, although reports of individuals with 2 nipples have been recorded. Male sugar gliders have a bifurcated penis to correspond with the 2 uteri of females. The age of sexual maturity in sugar gliders varies slightly between the males and females. Males reach maturity at 4 to 12 months of age, while females require from 8 to 12 months. In the wild, sugar gliders breed once or twice a year depending on the climate and habitat conditions, while they can breed multiple times a year in captivity as a result of consistent living conditions and proper diet. A sugar glider female gives birth to 19%, or 81%, babies per litter. The gestation period is 15 to 17 days, after which the tiny joey 0.2 grams, 0.0071 ounces will crawl into a mother's pouch for further development. They are born largely undeveloped and furless, with only the sense of smell being developed. The mother has a scent gland in the external marsupium to attract the sightless joeys from the uterus. Joeys have a continuous arch of cartilage in their shoulder girdle which disappears soon after birth. This supports the forelimbs, assisting the climb into the pouch. Young are completely contained in the pouch for 60 days after birth, wherein mammy provide nourishment during the remainder of development. Eyes first open around 80 days after birth, and young will leave the nest around 110 days after birth. By the time young are weaned, the thermoregulatory system is developed, and in conjunction with a large body size and thicker fur, they are able to regulate their own body temperature. Breeding is seasonal in Southeast Australia, with young only born in winter and spring, June to November. Further north in Arnhem Land, breeding is not seasonally restricted, and young may be born throughout the year. Unlike animals that move along the ground, the sugar glider and other gliding species produce fewer, but heavier, offspring per litter. This allows female sugar gliders to retain the ability to glide when pregnant. Socialization. Sugar gliders are highly social animals. They live in family groups or colonies consisting of up to 7 adults, plus the current season's young. Up to 4 age classes may exist within each group, although some sugar gliders are solitary, not belonging to a group. They engage in social grooming, which in addition to improving hygiene and health, helps bond the colony and establish group identity. Within social